Okay, so cancer is something that has historically been viewed as a genetic disease. And something that we've been learning more and more over the last several years is that cancer has some very significant metabolic components to it. So metabolism is very closely interwoven with metabolism, and metabolic adaptations and metabolic targets are an essential part of both how cancers develop and how we treat cancers. So just as a general overview, cancer is a really important thing for people in the developing world, or the developed world rather. It's a really important problem. It kills roughly a third of people, along with and along with heart disease. It's one of the leading causes of death in North America, Europe, and so. It's something like 50% of us are going to have a cancer at some point in our life, and it's killing something like 80,000 Canadians a year. So it's a really significant problem and something that we pay a lot of attention to. We worked very hard on solving here over the last many, many years. So one of the things that's we've known historically a long time ago about cancers is that they don't behave like normal cells. So is that and there's some reasons for that. So the cancer cells. So let's have a little bit of brief introduction. I guess we should have that here. Like what exactly are cancer cells? So cancer occurs when you have cells that are mutated in ways that are no longer behaving by the regulatory checkpoint. So most cells become quiescent after a certain number of cell divisions, and they shut themselves off. And this is to prevent damaged cells from replicating. So cancer cells didn't get the message, and so there are these regulatory checkpoints that they blow by, and they continue to grow and develop. So one of the impacts of that, I have a little diagram here. I guess I could draw it in. So normal cells will undergo a certain number of cell divisions, and then there will be a regulatory checkpoint that prevents them from going any further. So mutations that allow cells to continue to divide are called oncogenic mutations. Onco mean cancer, so cancer-causing mutations. And these are in a few. There are a few very common causes of them. P53 mutations in P53 they're linked to something like half of cancer mutations in P53, which is cell cycle checkpoint regulator. Other ones are tyrosine kinase linked. There are a few different drivers of cancer, but ultimately they all cause this unregulated cell division. So there's this sort of there's certainly a genetic element in mutations that enable this process to occur. But there's something else that goes on. So when cells stop behaving with regard to cell cycle regulation, they start to behave more and more like little pathogens, right? So the cell, as it divides, increasingly, is increasingly successful if it divides faster, if it becomes more efficient for converting carbon into biomass, and eventually if it finds new niches. And so what you see is that cancer cells originally they grow and they stay in place, and eventually they metastasize, which means that they change and they go elsewhere in the body. They move around in the body. Okay. So alterations in a number of metabolic pathways contribute to this developmental process, and the metabolic state that we see in these cells is really reflective of this sort of change, the shift in function from being a cell that supports the whole body to a cell that's just trying to promote its own gene pool, basically, right, and divide and grow quickly. So they really become much more like little pathogens as they grow and they divide. Yeah, let's talk a little bit about what happens here. So there are lots of metabolic changes that occur in cancer cells that differentiate them from normal quiescent cells or normal human cells. So a few of them, the classic one is the Warburg effect, which we'll talk about. Another one is that there's a increased reliance on glutamine metabolism, which we'll talk about. And we'll talk about some new emerging things that we're aware of, autophagy and albumin catabolism being one of those examples. So this is there are all kinds of things that can be altered, and we'll talk about a few of these. Yeah. So before we dive into this, does everybody clear about sort of the background about what cancer is, and you know, what does anybody need any more background on the cell biology or that sort of thing? Feel comfortable to get into? You feel comfortable for me to dive into metabolism? Clear to Michelle? Okay. So the Warburg effect was discovered by Otto Warburg. Oh, geez, I forgot now when this was. Is this 18 something or other, I believe, or I'll have to look that up. Was it when was that? Warburg. Sorry, I should have had that already. Otto Warburg. Oh no, wasn't that early. He was born in 1880, but I think he got the Nobel Prize in 1931. Anyway, so he described the Warburg effect, and the Warburg effect was that, you know, most cells will consume glucose and convert it into CO2, human cells we're talking about, when they are presented in oxygen-containing environments. But if you shift them to anoxic environments or microaerophilic environments, they'll increasingly use more and more of their glucose will be converted into lactate. So they use these anaerobic pathways. And so why is that, class? Why do cells use their anaerobic pathways under those conditions? Why do they make lactate? Class, to regenerate NAD, right? Yeah. So one of the peculiar things about cancer cells is that they will increasingly use lactate production regardless of whether oxygen is available or not. They don't care whether oxygen is available or not. All their glucose will get converted into lactate, and that's how they can generate their ATP. Now, this is really kind of peculiar if you think about it, right? As the cancer cells are dividing and doing all this stuff, then why would they use this anaerobic pathway, which only produces 4 moles of ATP per mole of glucose, whereas, oxidative phosphorylation is like 32 to 36 moles of ATP. So why do you think they would do that? Some ideas from the chat? Anoxic conditions inside a tumor. Well, yeah, Brandon, that's true. Yeah, tumors. So that's exactly right. So this is part of the discussion. So it's definitely true that oxygen selection for oxygen is an issue. Let's see if I can draw it here. So as the tumors grow, they become increasingly bigger and bigger and bigger. What happens is that the center of the tumors become very low in oxygen. That's because the cells are dividing a lot faster than they're getting supplied with nutrients. So the things are, when they're just a few cells away, it's not hard to get the oxygen or the nutrients. But as they get bigger and bigger, the oxygen concentrations get lower and lower. So if you look at a a gradient of oxygen concentrations, you'll find that the majority of oxygen, is just sort of is towards the outside of the tumors, and little smaller and smaller amounts on the inside. So there's definitely selection that occurs here in the center of the tumor for cells that are less dependent on oxygen. So that's one really good reason. Need to make loads of protein, right? So let's talk a little bit more about this. So the fact that cancer cells would need to tolerate anoxic conditions makes sense. But if you take a cancer cell out of the middle of this tumor and you place it in an environment where it has plenty of O2, it still makes lactate. So why do we do that? Yeah, that's exactly right, chat. This is quicker. It's more energetically efficient. So not efficient, it's kinetically faster. So one of the things that we can keep in mind is that so a couple of reasons for this. One is that the cancer cells are not evolving to be efficient with their energy in terms of using all the glucose well. They're just like the pathogens running around in your blood or whatever. They're trying to convert glucose into biomass as fast as they can, and they don't really care if it's inefficient. Do you remember how do malaria parasites make their energy? Where's the ATP come from in malaria parasites? You guys should know that now from your tutorials. From glycolysis. This is despite the fact they're living in a bath of glucose, they do exactly the same thing. They've actually, even though they have a TCA cycle and they're consuming glucose, they use glycolysis for all of their ATP, and they only use the TCA cycle to supply reducing equivalents for making nucleotides and that sort of thing, right? So cancer cells are kind of evolving along the same trajectory where, they may only be making 4 moles of ATP per glucose, but that really doesn't matter because the goal is to make energy fast, right? Right. And then the other point is that they're largely trying to expand. They're largely trying to expand their biomass, and their objective here isn't to use their incoming glucose very efficiently. Okay. So it may have some promoting effects on biomass. Okay. Okay, so that's the Warburg effect, which is classic. And then, so I've presented that as if it's all known, but actually the Warburg effect is a little more equivocal than as we presented it here. There may be some other things going on too behind the scenes, like it may be, it may have some links to regulation, cell cycle regulation in the cell. If you dig into the primary literature, you'll see that there's quite a bit of discussion about what exactly is going on with the Warburg effect. But certainly there's some selection for cells that are tolerant to low oxygen conditions. They have minimal need for being efficient with glucose, and it seems to be promoting anabolism. Those are sort of the main themes that we see from the Warburg effect. All right. So the other thing that happens a lot with cancer cells is they become increasingly reliant on glutamine catabolism, right? So this should seem probably less surprising to you now that we've done all this work with malaria parasites and other things, but like many cells don't use glucose as their primary carbon source, right? Fibroblasts from our cells, from our skin cells, for example, are very happy to catabolize glutamine, and a lot of other cells. So what happens in cancer cells, they increasingly will become better and better at catabolizing glutamine as a primary carbon source. And why do you think that this happens, chat? Why is this intuitive evolution of the cancer cell? Chat. Crickets. Glutamine is abundant. Need to use glucose and glycolysis to make ATP. So glutamine can add to TCA. Yeah, you can use it. Breakdown for muscle tissue. So we have a few different ideas why. Well, if we take a look at serum, like where does glucose come from, right? Need to make nucleotides. Where does glucose come from? Where is it? Where do cells get it from? Where do cancer cells get their glucose? From the blood, from the serum, right? So if we were to rank serum carbon sources, the first one here would be glucose. We'll talk about how much glucose is there in your blood. Do you know in millimolar? You should know this now from your malaria projects. Assuming you're not diabetic, what is it? Chat, hello. How much glucose? A kilogram per mil? 1 femtogram per mole? What is it? 5 to 7 millimolar. Okay, somebody was Google searching, I guess. Yeah, you're right. It's 5 millimolar, roughly. So what do you think the second most abundant carbon source is in your blood? Nice. Right. 5 millimolar is exactly right. Yeah, it's in like the 3 to 10. Depends on whether you just had a blood meal. Somebody who's diabetic, it could be quite a bit more. What do you think the second most abundant carbon source is? It turns out the second most abundant carbon source is glutamine in your blood. No, not carbs. Glutamine. We're talking about things in the serum. And what do you think the third most abundant carbon source in the blood? You're probably not going to know this. The third one is albumin. You guys know what albumin is? We'll talk about it more in a bit, but it's a protein that carries fat. It carries fatty acids around for you. It also carries drugs and greasy things around. It's a transport protein that carries fat. It's the third most abundant carbon source in your blood. Okay, so glutamine metabolism is something that changes a lot in cancer cells. Oh yeah, why does this make sense? Well, because if we go back to our evolution of the cancer, as it gets bigger and bigger, it goes through this stage where you have a O2 gradient. What do you think happens to glucose as your Warburg effect driven cancers proliferate? What do you think happens to glucose concentrations at the center of the cell? Chat. It goes down, right? They decrease. So same thing happens. O2 goes down as you go from center of the cell out, and glucose concentrations also start to go down. And so the cells are increasingly reliant on their glutamine as a carbon source, right? So glutamine comes in here, and you guys should be familiar with this now, how glutamine is converted into glutamate, and that can be converted into alpha-ketoglutarate, and that can go into the TCA cycle, or all kinds of other biosynthetic pathways. The other thing that is, I guess, is useful to do is that it's participates in glutathione biosynthesis, its biosynthesis there is one of the ending products here for the cell. So if you map glutamine catabolism by, I think these were isotope labeled glutamine, and then they map glutamine catabolism and they did this imaging to find out where all the glutamine catabolism was done. Where the source and sink cells are. This is what it looks like in your body. Is oxidative stress a big deal for cancer cells? It's a big deal for proliferating cells. Oxidative stress is, yeah, in general. Of course, is oxidative big stress, is that a big deal when you're living at the center of an anoxic tumor? Yeah, not really. Probably not. Yeah, I'm not actually sure why this slide's in here. Like it is an issue for oxidative or cells that are growing in oxidative environments. Glutamine can go into glutathione biosynthesis. I don't know how much of a role glutathione is in, or glutathione catabolism is in cancer though. So there are a handful of cells that end up being big glutamine producers and others that are big glutamine consumers, and that pattern changes when you are in cancer state. So your liver increasingly takes a larger and larger role on producing these and supplying it to the rest of the body. Yeah. And you'll find these hot spots here where glutamine is taken up and consumed, and that sort of puts everything else as everything else into production mode. So this selection for glutamine catabolism ultimately results in cells that, similar to the Warburg effect, become addicted to glutamine. So the Warburg cells are addicted to glycolysis, and then some other cells can become addicted to glutamine. And so what this means is that you get to the point there that they are totally dependent on glutamine catabolism, and if you interfere with other pathways, they will die. So this has made some folks really interested in investigating some of those glutaminase and those other enzymes we were talking about as potential metabolic targets for cancer drugs. So the third most abundant carbon source that we talked about in the blood is albumin. So albumin's a part of this, the serum fraction here. It's if you take the plasma or serum and you heat it up, it'll turn into something that looks like an egg white. That's because egg white albumin and serum albumin are related. They have similar functions. They store fat. They keep them in the blood. They don't carry triglycerides around. They carry around either free fatty acids or they'll carry around things that are drug-like. It's a way of getting molecules that aren't soluble in water to move around in the blood, move them from tissue to tissue. So that's how we move them around. Well, it turns out it's also a very abundant carbon source that's accessible to cancer cells. And so cancer cells will, and this is just newly emerging literature here, is stuff that's being done by some folks that I did my postdoc with. So this is Yuri Comforst and Michael Nofel and Sean Hackett. These guys were doing this research during my postdoc. But what they showed is that if you isotope label if you use these isotope tracing experiments, you can actually map the uptake and use of albumin as an amino acid source for cells, and they start catabolizing it and using the albumin as an amino acid source. And then this is a significant contributor to their growth. And also, so in this experiment, they did a few different experiments. One that shows the levels of amino acids inside the cells with and without the addition of BSA, and then they show that with and without the inhibition of a lysosomal processing step. And so they show that these have a direct impact on uptake of the amino acids from the albumin. So, and this is part of sort of a wider category of activities called autophagy. Well, autophagy means self-eating, autophagy, right? And so glutamine catabolism and other forms of autophagy, breaking down proteins to feed cell metabolism. This is a really hot new area of cancer research right now. So as it turns out, is also a really active change that cancer cells undergo. So this is sort of a map of all of the metabolic activities now that are being thought of that are going on in cancer cells. So there are just changes in glycolysis to the Warburg effect that we were talking about earlier. There are these uptake and increased utilization of glutamine that we've talked about. There's some emerging literature about changes in scavenging and lipids. There's lots of changes here in scavenging of albumin and lysosomal processing, and then there's also active processes going on inside the body where they're breaking down things that inside the cells that they don't seem to need, and via autophagy. And all this is ultimately resulting in some very significant changes inside the cell. There's also lots of changes in the regulation of metabolic genes. And then any of these things could potentially be targets here for new cancer therapies. So why do we care about all these metabolic changes? Because ultimately, these are the things that we can target with new anti-cancer drugs. So that leads us into this next section. How do the drugs work that we currently have, and what's the emerging frontier look like? So we'll talk about some things that are emerging for targeting glutamine catabolism. We'll talk about some ribonucleotide reductase inhibitors, and we'll talk about DNA-oriented kinds of drugs like methotrexate. One of the hallmarks of the cancer cells that we're just talking about that's not currently drugged, but something that could be that could potentially be useful are these inhibitors of glutamine catabolism we just talked about. So there's some discussion about inhibiting glutamine uptake or transport, inhibiting the essential processing steps like glutaminase or glutamate dehydrogenase, things that prevent the carbon from glutamine from getting into the TCA cycle. What do you guys think about these categories of drugs? Are these a good idea, a bad idea? Like what do you expect would be an impact if you had a, say, glutamine uptake inhibitor? Is this a good idea or not? Chat. Would this affect the urea cycle? Yeah. I mean, would inhibiting glutamine catabolism affect the urea cycle? Yes, in the sense that there would be less ammonium going into it, I think. Let's see, might this be bad for cells that actively prefer glutamine as a carbon source? Yeah, it would be problematic for things like fibroblasts and some other cells. Yeah. What else? Wouldn't the cell switch into another source of carbon? The cancer cells? Yeah, they probably would over time, although some of the literature, like these glutamine-addicted cancers and these, make it seem like they're not tolerant to switches over time. It may be that they accumulate enough mutations in other pathways. You know, the way evolution tends to work in cells is that deleterious mutations cause the populations to go down, but ones that are neutral are preserved. So over time, if you don't really need a pathway, then you can lose it, right, with no deleterious effect. So the idea may be that cancer cells are evolved in ways that make them unable to use other sources, and then they would be more dependent on it. But like, as you probably have guessed, if you add a glutamine uptake inhibitor, they'd probably start evolving significant pressure to find other metabolic routes, right? So, I mean, this inhibitor has the same problem that all kinds of inhibitors of cancer have, which is that ultimately you're trying to treat something that is one of your body's cells. And so when you treat one of your body's, you're trying to inhibit a process in the body, it's toxic, right? So all cancer therapies currently are toxic in one way or another. And then the principle that they're working on is that are more toxic to rapidly dividing cells than they are to non-dividing cells. So since your quiescent cells are not making new biomass, they're more tolerant and able to deal with starvation for glutamine than your cancer cells are. And that's the that's the foundation for this theory. Yeah, does that make sense? So that same concept applies to the nucleotides as well. One of the really key distinctions between quiescent cells and proliferative cells is that proliferative cells have to make DNA, and so cancer drugs. There's many successful cancer drugs that ultimately function in one way or another by inhibiting DNA biosynthesis, because that's something that's, as we talked about when we were talking about this in our last lecture, something that normal cells are not making DNA normally, right? You have one copy, you got your copy of the DNA already in the cell, and as long as you're not dividing, you're not making any. So it's something that's peculiar that cancer cells are doing. They're definitely making DNA, and so things that inhibit that will selectively kill the cancer cells. Unfortunately, it kills any proliferative cell in your body, so that's a problem for hair cells and immune cells and quite a number of other cells that, skin cells that are dividing as a part of our normal function. So that's why people on chemotherapy will end up losing their hair and have this kinds of changes. It's having a big impact on all the proliferative cells in the body. But they're both. Can you just close your presentation? It's locked. There are quite a number of. Here we go. Sorry. There are quite a number of drugs that treat these pathways. So two of them here are shown here. So we have chlorophabine and fluoridabine, and those guys are both acting as purine analogs. So they look like purines, they go in, but they're suicide inhibitors or interfere with the enzymes in other ways. They have these chloro-fluoro substitutions that make them problematic. So another classic cancer drug is 5-fluorouracil. This is used in quite a number of systems where you want to inhibit DNA replication. So this step ultimately prevents or inhibits this thymidine thymidinolate synthase reaction. This is the one we talked about very recently here. And there's this methyl transfer reaction that has to undergo from this methyl tetrahydrofolate reaction to this dihydrofolate analog. And so this inhibitor here sits at that site and then prevents that transition from happening, right? So it's sitting in here. I think it becomes a suicide inhibitor here in this enzyme. Anyway, so this ultimately, it's a suicide inhibitor. So it stays inside the enzyme, prevents the replication of DNA, and it's used a lot. It's used for treating colonic, esophageal, stomach, pancreatic, breast cancer, all kinds of things. 5-FU, and see what else do we have? Methotrexate. So methotrexate is one of the most commonly used anti-cancer drugs. It also inhibits essentially the same theme, which is that it inhibits the recycling of tetrahydrofolate. So if you recall from our last lecture, so that methyl transfer reaction then has to undergo regeneration that involves going through this DHFR, and which dihydrofolate reductase reaction. And so this is both a target for malaria parasites, and it's also a target for cancer drugs. So methotrexate inhibits this pathway and prevents the recycling of tetrahydrofolate. And so that also ultimately functions by inhibiting DNA replication, because the methyl tetrahydrofolate synthase reaction stops. Yep. So is everybody comfortable with I guess the general themes here, that cancer cells have a lot of metabolic changes, and they undergo some metabolic activities like rapid proliferation that are different from regular cells. And so the approach of all cancer therapies is to find pathways or processes that are unique to the cancer cell, or at least more enriched in the cancer cell, so that when you treat them with it, they selectively they have a more significant impact on the cancer cells than they do on the healthy cells. Yeah. So, so this is not the end of what we know about cancer. So right now we're in a really interesting new frontier where metabolomics tools are being used increasingly to understand how cancer works at the molecular level. And there are all kinds of strange new emerging observations that we have about cancer that may ultimately lead to either new diagnostic strategies or new ways of controlling cancer. So this is a really famous paper in the metabolomics literature that this came out of. It's one example of how metabolomics screening tools have identified new potential ways for supporting early diagnosis of things like prostate cancer. So this particular study was the objective of it was that they're screening serum samples from patients, and they're looking for early markers of prostate cancer. So currently, prostate cancer has this issue in that it is generally not diagnosed or known until it's progressed and it's fairly serious. And so early diagnostic tools for prostate cancer are really important. And also prostate screening is really unpleasant. So it's something that where compliance is low, people don't really want to do it. But if they don't, then, by the time tumors are detected, they're really, it's generally pretty far along. Well, there are a few screening tools where you can look for things in serum, and so there've been quite a lot of interest in finding things that just allow you to do a blood sample or something like that, and which would potentially allow a wider reach for screening applications. So this is the one of those studies that was doing that, and then so these samples were taken from patients who were either they took tissue out of regions from patients that were either benign or they had this prostate cancer or metastatic prostate cancer, and then they looked at the metabolism from these different lines and looked for things that differentiated them from normal cells. So using this metabolomics approach, so really similar to what you guys are doing in the lab right now in our tutorials. One of the things that they found that was very different is this molecule called sarcosine. And so they published quite a number of things, and they found that this levels of sarcosine seemed to be very predictive of the metastatic cancers. This was published in Nature back in 2009. So what is sarcosine? So sarcosine is a derivative of glycine. So it's made from this directly from it's made from glycine in one step, and then it's converted back into glycine with the sarcosine dehydrogenase reaction. So what they did was they showed that by knocking down either the enzymes involved in making sarcosine or degrading sarcosine, they can modulate the invasion and of cancer cells, and so that's what's showing here is that by using siRNA to knock down the cells here or knock down this biosynthetic enzyme, they can modulate the invasion efficiency of the cancers. Similarly, they can show that by knocking down this sarcosine dehydrogenase enzyme, they can modulate the activities or the concentrations for the sarcosine dehydrogenase. So the theory here was that they were looking for looking for new biomarkers. They found the sarcosine. They're finding the metabolic pathway that's ultimately affecting it, and they're showing that it does appear to have some direct impact. Modulating this pathway does seem to have some direct impact on the how aggressive the cancers are. Okay, so I think we had just sort of a quick tour of cancer today. I think some of the themes we talked about, main themes you should know about in general, are the Warburg effect, that there's glutamine catabolism, and that autophagy, I would say, albumin and autophagy in general are three emerging themes that cancer cells are known to do that contribute either to their progression or their metastasis. That many of the drugs that we have work by inhibiting one of these processes or other metabolic processes that are unique to cells. And then we talked about an example of how biomarker screening is also inherently related to metabolism. So the ways we can do rapid diagnostics now are ultimately related to metabolism. So what's the theme here? The pathways that you guys are learning in this class are absolutely essential to understanding why cancer kills 30% of Canadians, and ultimately could play a direct role in diagnosing a cancer in you. 50-50 chance, I guess, at some point in your life that you'll have a cancer. So these pathways that we're learning about aren't arbitrary. I guess is one of the points I wanted to make today, is that all the things we're learning, they're tough, and like we have many steps, and it's hard to learn. It's hard to keep them all. It takes a lot of work. That's worth knowing. Like, understanding these pathways tells you about the basis of life. It tells you about why life goes wrong, and it ultimately gives you the tools to make life better for people. So I think that's one of the take-home messages from today, is that these themes that we're working about, like, these are the vocabulary on which we're painting, the picture of, future medical sciences on. Anyway, that's all I have for today. Happy to chat more if anybody wants to discuss metabolism and cancer themes. Thank you, Brandon. Thank you, Kosar. Thanks, guys. Thank you. I'm glad you liked it. Is there a reason why cancer cells is a bigger deal in developed countries? Yeah, Jeremiah, there it is. The reason why is because people in developing countries die before they get cancer. So in developing countries, a third of people die from diarrhea. And they're much higher infant mortality. There are some really stark differences between how people live in places like Canada and the US and Europe, and how people live in places like rural Peru or sub-Saharan Africa, or Papua New Guinea, and that extends to how people die, has a big impact on how people die. So one of the luxuries and curses of living in a developed world is that we have these chronic diseases that become a more increasingly significant burden on our healthcare. So, Tarek, does this have to do anything with highly processed foods or developed countries? Probably not. I don't think so. I think it's mostly reflects the fact that when you live, like, you have to die of something, right? And so the things that you die of, if you don't die of an infection, and you live out all of your possible potential, are going to be heart disease, the various changes in your brain, so all Alzheimer's or Parkinson's or these sorts of things, or or other kinds of diabetes, heart disease, metabolic syndrome. Like these are the things that ultimately kill us if we don't die from infectious diseases. These are just the defects of humans, right? And so I think there's certainly things you can do to modulate your metabolism. Oh, geez, we didn't even talk about that. How you can modulate your diet to extend your lifespan. We could do that sometime. We talk about that a little bit too. But caloric restriction is the only thing that we know that will expand your lifespan. And part of that is limiting long-term exposure to reactive oxygen species and preventing your cells from doing too many divisions. Yeah. Yep. Right, makes sense. So if people in developing countries could live longer, would they see similar rates of cancer? Probably, yeah. There are definitely some trends in diseases that are linked to places of origin. For example, certain parts of the world, people are more prone to obesity and diabetes. There's some theories behind that. The thrifty gene hypothesis, which is that people who are originating from areas where there was a lot of periodic starvation have genes that have been selected to help encourage and promote the fat storage, and those same kinds of genes ultimately promote diabetes. So certain people from certain parts of the world are more likely to die from diabetes, whereas people from other parts of the world may be more likely to die from cancer. There's certainly regionally based risk factors for the various diseases, but yeah, if people live longer, they're going to die from something, and cancer is very likely going to be one of them. Sarcosine levels used more often in cancer diagnosis tests now, or that, yeah. Well, so there's some back history on that sarcosine thing. It's not used at all as a diagnostic test. There is another thing that is used as a diagnostic test, which is prostate-specific antigen, PSA, and it's really crappy. Its predictive power is so bad that it's like the problem with PSA is that it will find tumors in people. It will find tumors that are, prostate cancers grow very, very slowly over a very long period of time. And so it will find those tumors in people who might never know that they had the cancer and will die long before the cancers progress. And so there's some discussion about whether PSA is good to screen for or not. The sarcosine project, while it looked really good in the original trials, it doesn't, it's kind of equivocal right now. People are on the fence about whether it's better than PSA or not for screening. It's currently not used as a screening tool though. But it was a promising avenue. You're welcome. Let's see. Okay, guys, I think we're up on time. I hope you enjoyed the lecture.